Hi everybody, my name is Lauren Henry and I made this video very slapdash last night because, to be perfectly honest, I forgot I said I was going to do it, but I did want to tell you guys about my time at Woods Hole Oceanographic Institute as a co-op. I was there in the January through June of 2013 and I worked in the Anderson Harmful Algal Blooms Lab. I was the co-op underneath Mindy Ricklin, and I did a whole lot of genetics with Alexandrium fundiense, which is a harmful algal bloom. So this here is the lab on a very nice day when we were there. This is Redfield Laboratories in Cape Cod in Woods Hole. This was the inside of the lab that was sort of facing my lab station and all of the reagents and freezer stuff that I used. I really spent most of my time pipetting, but it was fantastic. I learned how to do a whole lot of genetic techniques such as PCR, riff flips. I learned how to do transformations, restriction endonucleases. It was just all kinds of crazy technical terms you all probably heard about in genetics, but honestly, doing it in a lab setting made it very real for me, and it really beefed up my resume, which was all what co-op is about. And it really put a ton of experience I didn't have onto my resume, which made it really valuable for me. So a big focus at the Anderson lab was on harmful algal blooms, specifically the dinoflagellate Alexandrium fundiense. A lot of work that I did on Alexandrium was taking DNA from different samples collected throughout the course of a harmful algal bloom and seeing if there was any sort of real difference in the DNA and population genetics as the bloom progressed, and which ones were more toxic versus which ones were less toxic. And it was very interesting to come up with absolutely no data, because that is just what science is sometimes. But that led into the other half of what I did at Whoi, which was studying a couple other different types of harmful algal bloom. So my supervisor, my PI, Mindy Ricklin, was actually doing a lot of work on Gambierdiscus and its different species. What I did a lot of was either looking at samples and doing counts of Gambierdiscus density, I guess, in the sample. So I would basically take I'd shake up this bottle of sand and I would put a small sample of it on a slide and I would scan it and count how many Gambierdiscus were in that set volume, and then we could kind of extrapolate up from there to see just how many Gambierdiscus were in that particular area, that sample size that they collected from in St. Thomas when they were down doing field work. And I know that sounds kind of easy. I mean, it's a fairly cute looking little dinoflagellate, and you guys have been looking at it for a good like 30 seconds, but unfortunately, there's a lot of other dinoflagellates that can be found in samples in St. Thomas, so I held Gambierdiscus. I had to tell it apart from this here Ostreopsis, and couldn't get them confused with Pro or Centrum either, so there was a whole lot of similar things. You got a really good eye for it. This was just a really cool foraminiferan I found in my sample. I thought it looked like a zombie brain. Now, when I wasn't doing my basic chores, my usual stuff around the lab, I actually had a couple cool projects. The more I proved that I was fairly competent at working alone, the more freedom my supervisor Mindy gave me, and it was really fantastic because she actually let me spearhead a couple really cool projects we did. She had gotten these mud samples from a collaborator all the way from China, and due to some funny import restrictions, they had been sitting in a warehouse for about 6 months in the dark, so we just had no hope for them. But the cool thing about these Alexandrium fundiense dinoflagellates is that when conditions are bad, they go into a cyst form. So we went through all of this mud and we sifted through it underneath a microscope, and I actually was a bad scientist, but this is how you do it. I would pick up individual cysts using a capillary tube that we actually melted out over a flame and stretched to make it even tinier. And we would pick up individual cysts, so like individual cells underneath a microscope. And I got really good at mouth pipetting, which is like the thing you're not supposed to do, but when you're just sucking up seawater, it's not particularly dangerous. And I ended up isolating 10 96 well plates. I put one cyst in every single well, and then we put them in the incubator, and I would just periodically check them to see if any of the cysts had germinated into vegetative cells, which are the ones that swim around. And as it turns out, it was an unprecedented success. We did not anticipate how many of them would germinate despite their rough treatment. And we actually ended up founding, I think it was 48 clonal cultures. So 48 cultures that were just entirely one DNA type from one exact little cell. So they were all exactly the same DNA. They were clones of each other. And then I think it was like another 142 non-clonal cultures. So we just had a ridiculous amount of new cultures, so much to the point where they actually bought a new $10,000 incubator just for all of my samples. It was really, really fantastic. The great thing about Woods Hole Oceanographic Institute and Harmful Algal Blooms Lab is that they have a lot of funding and you get used to nice things. I was once complaining that my multi-channel pipette didn't really do a good job anymore, and they just bought me a new one, and just because I liked it, they bought it in pink. So it's really fun how flexible they are there, and made me feel silly, but I loved it. And not to give you guys the impression it's all lab work, I actually did spend a good amount of time out in the field. Mondays were field days. For me, we did all of our like giant seasonal work on the Mondays. Maddie and Adam, who were the other two co-ops, they were out in the field a lot more frequently than I was. Adam got up at like 5 every single morning to go drive 2 hours out to the Nauset field site where we did all of the work. And props to him, because I don't think I could stand on a salt flat at 5 in the morning in a pair of waders in the middle of February in Massachusetts. But as you can see, we had a lot of fun wearing our little flotation devices with our environmental sample processors. Another great part about working at Woods Hole Oceanographic Institute was that there were a lot of cool things that were just happening around there. Isaac just happened to walk through a lab one day and realized that they were rebuilding Alvin, which was the submersible that was used to find the Titanic. But on this particular day, we were all super excited because James Cameron himself had shown up to donate Deep Sea Challenger. So here's the man himself giving the speech as he was donating the submersible to us. And say what you will about him being a movie director or a butthead, but he had some really fascinating things to say about being to the bottom of the ocean and seeing all of these cool new things, and his genuine enthusiasm and really surprising amount of knowledge about all of the things he found down there. It was really cool. It made you feel kind of inspired that this big Hollywood producer could contribute so much to a field, and it was just really exciting and really cool. Aside from the really cool opportunities that working at Woods Hole has afforded, it was actually just really fantastic to live on Cape Cod for 6 months. There is not a whole lot to do on Cape Cod. It is kind of what you make it, so it's really important to get to know the other co-ops before you get there. We actually all rented a house together, and it was a whole lot of fun, and we had a really fantastic time. Every weekend was an adventure. If we needed to go into the city, both Adam and Isaac had brought their cars, but if not, Maddie and I could just take a bus, because there's one that runs from South Station straight to Redfield Laboratories and back, so it's literally a 2-hour bus ride, and it's not too expensive in the winter. A couple of the cool things that were to do around town were the obvious quaint New England things. Maddie and I took a day trip over to Martha's Vineyard and rode our bikes around the island. We got lobsters one time from the farmer's market, and we were able to do a giant lobster cookout with all of our friends. If it was just a slow weekend and we just had some fun. We were all 21 when this picture was taken, by the way. But this was our house in Falmouth. This was 1 Wilson Road. And this was our house after a giant snowstorm, where we didn't lose power, but we basically we assumed that position on the couch for 3 straight days and just watched a ton of Disney movies. So before I paint the entire thing as idyllic, you do have to realize that you might be there during winter. That being said, when summer rolled around, it was really fantastic. We were at the beach almost every single weekend. We had an unseasonably warm May and June, so we had just a really fantastic time. We got to explore some of Cape Cod's really awesome natural wildlife, like these cute seahorses. Actually, one of my favorite memories that I made at Woods Hole was when Maddie and I went to Martha's Vineyard on the ferry for our bicycle trip, and I had just bought my bicycle and the weather was finally warm enough. We were super excited, so we went out on this dock and I put down the kickstand. It was blowing really hard, and as you guys can see, my bike kind of vanished. And we were able to fish it out of the water, but for a while there I thought I was going to have to strip down and dive off a dock to recover my bike in the middle of March in Cape Cod. So you can understand I wasn't thrilled. Thankfully, a very nice man helped us fish it out with a broom that was in the back of his car, but my bike never really recovered after that. Overall, though, I had a really just amazingly fantastic time at Woods Hole, and I am now best friends with my old PI, Mindy Ricklin. She still sends me different kinds of job offers. She drums up for me, and we just have a really great relationship. We'll send each other cat videos, and it's just amazing to have that networking opportunity, and I just look back on that time super fondly, and it was amazing. And if you guys have any more questions, feel free to email me at misslaurenhenry@gmail.com, ms, and then laurenhenry@gmail.com, or henry.laur, L-A-U-R, @husky.neu.edu. So I hope you guys enjoyed, and I hope you have fun picking out your co-ops. Bye!